It's 4:30 and that means it's lightning talk time. So I know that we have a few new faces in the crowd this afternoon. So I want to make sure that I tell everyone how this is going to work. And also I know that we'll have a couple more people kind of join us as we get going. And so we are going to hear 3 lightning talks, which means rapid succession. Each speaker will have 10 minutes to present their slides and their research. We will hear from Yuan first, and then Kiandokht, and then Tian. They will present. We will ask you to hold your questions, and then we will open breakout rooms associated with each speaker afterwards, so that you can go in and interact with the different speakers. You will be able to kind of navigate, ask your questions to Kiandokht, for example, and then navigate to Tian, and then wrap it up with Yuan, or in any order that you like. So that is roughly how the process will work. We are recording all of these presentations. We will ask all of our speakers for permission before we post them all. So just a note that we are recording, and permission will be coming to the 3 of you if it has not already, so that we can share more broadly. But we will be sharing this in the context of the conference, if that is okay with everyone. Excellent. Okay, so without further ado, I believe, Yuan, you are up first if you would like to get your slides ready.
Can everybody hear me now?
They look great, and we can hear you just fine.
All right. My name is Yuan Ren. I'm a postdoc from the lab of Julian Barrow at Yale University. I would like to thank the organizers for giving me this opportunity to share what we have been doing in the lab. So in this talk, I'm going to introduce a new set of force sensors that we use to measure force in endocytosis in patient yeast. So first, some background. So this is a simplified version of endocytosis. You have on top, outside the cells, and then this is the plasma membrane. And on the bottom is the inside of the cell. You usually have some materials. Well, the cell usually internalizes materials using different pathways, but unless this is water or something really small, you need endocytosis for that. During this process, you use a lot of proteins to generate an imagination on this membrane, and eventually to form a vesicle that encloses all the materials. And then what's inside is transported into all other organelles for utilization, or sometimes transported back in another process called exocytosis. So if we go a little more deeper into the details, we can have this. This is a snapshot of different stages during endocytosis. And in many cells, especially in patient yeast cells, this temporal evolution has been studied really well. And we know already that they follow a very strict timeline. For example, if you start bending the membrane at, the membrane at 8 seconds before the pinch off to form a vesicle. And in this process, a lot of proteins are needed because there are a lot of forces you need to overcome. So for example, you need to overcome the turgor pressure, the bending rigidity of the membrane, and also the membrane tension. So especially in patient yeast where the turgor pressure is very high, you absolutely need the polymerization of actin to generate enough force for the internalization of the membrane. So here at the bottom half, you have the actin polymerizing, and you need also an endocytic coat, which is a mixture of different proteins that comes and localizes to the appropriate positions to connect the membrane to the actin, so that the forces produced by the actin can be transmitted to bend the membrane. So in this process, actually, the modeling is a little bit ahead of the experimental approaches, because to be able to measure the force in this transient and small place is very difficult, especially force measurement in vivo is not easily done. So we would like to develop a new force sensor so that we can know how much force is actually being transmitted from actin to bend the membrane. And the protein we are focusing on in this project is called N4 in fission yeast. So it has a domain at the N terminus that binds to lipid and has an actin binding domain at the C terminus. So if you look in this diagram, you can see how it is anchored at the head to the membrane, and then at the leg, it's binding to the actin, and it directly connects the actin to the lipid and directly transmits the force from the actin polymerization to the lipid. Also in this dotted line, you can see the Clathrin coat. So this is a really long molecule. So in order to measure force, what you need to do is to insert a force sensor somewhere here before the actin binding domain, so that the force on this protein can be measured if you have a good readout. So we are not happy with the current sensors, which is based on Fred, because the Fred sensors are typically big, and the insertion itself has some effect on the function of the protein during endocytosis. And also Fred is based on a very sophisticated reading, and it relies a lot on the equipment and careful interpretation. So the approach we are thinking of is to use a protein motif that is very commonly appearing in the proteins called coiled-coil. So two RB helices can form a coiled-coil, and in this conformation, they bury their hydrophobic residues in the middle, and they have their hydrophilic residue on the outside. So you can actually pull on the two ends of the RB helices. You can pull here, and then you can unfold this coiled-coil using force. So what's useful is for each coiled-coil, it has a signature force threshold below which nothing happens, but above that force threshold, this ruptures and the hydrophobic cores will be exposed. So this can be done in vitro using optical tweezers if you attach this motif to two beads. And you can measure this by pulling on it and then relaxing. And then for one molecule in vitro, you can unfold it at a certain force threshold. And if you relieve the force, it can refold reversibly for a million times or until you get bored. So for this first coiled-coil, from this measurement, we can see that the force threshold from the distribution is about 11 piconewton. So this we are thinking, we are calling CC 11 piconewton because it is a force sensor at 11 piconewton. So what it means is if the force acting on this coiled-coil is below 11 piconewton, nothing happens. But if it's above 11 piconewton, it unfolds. And what's interesting is in the in vivo situation, especially if you are inserting this into a dimer, is the refolding will sometimes they will refold onto other molecules. So you can think of this during the refolding process. The RB helix from one molecule has a chance of interacting with the RB helix from another coiled-coil, and then this will lead to the formation of intermolecular interactions and eventually protein condensations. And this is exactly what we observed after we insert the coiled-coil that unfolds at 11 piconewton before the actin binding domain of this protein. We see the formation of big protein condensates that can be easily detected, and it's very different from the natural endocytic patches you see. So this is normal protein localization. So in the cells, it's pretty dim and small. After you insert the CC11 piconewton, you'll see the appearance of big protein droplets indicative of a local force here before 11 piconewton, above 11 piconewton. You can do a positive control where you shorten the linker to force the coiled-coil to be open, and then the condensates are bigger and brighter. So what is good is this is not some special feature of that particular coiled-coil, but seems to be a general feature of all the coiled-coils. So we designed more. So here we are showing 4 coiled-coils, each opening at a different force threshold. We have here from 8 to 20. So each of those coiled-coil force sensors are asking one question when they are inserted into the protein. And the question is the force above or below my force threshold? So we can insert the 4 coiled-coils into 3 different positions of N4. And you can see aggregations at some cases, but not all cases. And the boundary is the force range on that at that place. So using this force sensor, we are able to detect different levels of force on this N4 molecule. It's between 11 to 20 here before the active binding domain, and about 8 to 10 here and 10 to 11 here. And this is different from what people commonly think about the forced transmission during endocytosis. What we think is happening that can cause this difference in force is the binding between other proteins. The binding of other proteins 2 and 4 is helping the transmission force during endocytosis. I'll be happy to talk more about this afterwards. So I would like to acknowledge the funding agency, and also this is a collaboration project between the Barrow Lab and the Zhang Lab. Thank you all for your attention.
Thank you so much. That was a wonderful presentation. And my screen just got stuck in a not very pleasant spot. Next up, we will hear from Tian.
Thank you very much. I'm going to share my screen.
It looks good and sounds good.
Thank you so much. Thank you for the opportunity for me to share our research with you. Today, I'm going to talk to you about cell lineage specification at developing tissue boundary. Specifically, we're interested in 2 types of motor neurons that are differentiated during the development of mouse spinal cord. So as the schematic shows here, these 2 types of motor neurons marked by 2 transcription factors, HOXA5 and C8, and they form a sharp boundary along this rostral-caudal axis of the organism, so basically head to tail. And I should mention that this project is in collaboration with my long-term collaborator, Yuan Ren. And most of the experiments that I'm going to show you were performed by a previous graduate student in Chen Lab, Zhongrong Li. And this is one of the experiments that inspired us. When we stained these 2 proteins during the spinal cord development, we found that the lineage segregation of these 2 types of motor neurons occurs sometime before the embryonic day 12.5. So before that point, some of the cell express both transcription factors, but at E12.5 and onwards, the lineage segregation is very clear and there's a sharp boundary along the rostral-caudal axis. The question is, how do these cells make robust lineage decisions? It's an important physiological problem because these motor neurons, upon the upregulation of these factors, will further specify these subtypes and eventually innervate different muscle cells. And the upregulation of these 2 genes are influenced by the 2 morphogen signals, retinoic acid and fibroblast growth factors. These 2 diffusive molecules will form shallow and antiparallel morphogen gradients. But we know these signals are typically noisy and they compete at the tissue boundary. And more importantly, they are transient. So how do cells make robust decisions in this challenging environment? There is this classical model that explains this tissue boundary formation, which is that 2 transcription factors have this cross-repression relationship. And it's been used to explain tissue boundaries at multiple locations of Drosophila embryo. And this circuit is found in mammalian development as well. So we're looking here at the neuron tube along the dorsal-ventral axis. The question is, does this circuit explain the tissue boundary between A5 and C8 cells? And to explain this possibility with a model, we built this hypothetical transcription cross-repression model based on this cross-repression relationship. And with this simplified reaction diffusion system, we assume that there is this distribution of these morphogen molecules across the rostral-caudal axis. And with the help of the cross-repression gene network, all the cells in this grid system decide their fate in a robust manner. And there's a sharp boundary formed between these two regions. But does this circuit exist in these cells? So we use mouse embryonic stem cells to look at the relationship between these two genes, and we verified that A5 is inhibited by C8. But it turns out the opposite relationship is not true. So the upregulation of A5 does not influence the expression of C8 in these differentiated motor neurons. But the question is now, does this experimentally validated network can explain the lineage segregation? So we built this transcriptional unilateral repression model, and it turns out under the same condition, this model cannot explain the lineage segregation at the tissue boundary. And specifically, a lot of cells have this hybrid phenotype at the boundary, and the phenotype is fluctuating. So both models were falsified, but in terms of performance, what causes the difference between the two models? So in terms of the dynamical system, the cross-repression feedback loop creates these two attractors, and this is a phenomenon called bistability, and the two attractors can lock this cell phenotype into their own lineage. So that helps the lineage segregation at the tissue boundary. But with the loss of the feedback loop, the system is not bistable, and the phenotype is sensitive to the fluctuations of the microenvironment. So that explains the mechanism of this TCR model. So we wondered maybe the model can be used in a more generalized way. There may be two factors upstream of these two molecules that are mutually inhibiting each other. But if this generalized TCR model is true, one would expect that the distributions of the mRNAs of these two genes have sharp boundary. At least they should be bimodal. But it turns out this is not true when we look at the data of in situ hybridization and single cell RNA sequencing data, which revealed that a lot of cells express both factors in terms of their mRNA. So how can the system achieve robust lineage segregation without a segregation of their mRNA? So we looked into the post-transcriptional regulation, and specifically, we incorporated two microRNAs that inhibit A5 and C8 respectively, and they are miR27 and miR196. And when we incorporated these two microRNAs into the model, we found that this new experimentally validated model explained the difference between protein and mRNA distributions, but the boundary is still fuzzy. This is due to, again, the lack of feedback loop. And we further excluded several other models, which I don't have time for, but I should mention that these conclusions are not sensitive to the choice of parameters, so they are valid in a wide range of parameters. So we then hypothesized that there might be some hidden feedback in this reaction network of mRNA and microRNA that cannot be described with these coarse-grained models, which primarily based on Hill functions. So, for example, when we built a generic model of mRNA microRNA interaction with mass action kinetics, we found that, interestingly, this little model with an mRNA with two binding sites of for the microRNA, there's a wide range of parameters that allow bistability, which could be important for the lineage segregation. And through some mathematical analysis, we found that there is an interesting relationship among the degradation rate constants that allow bistability. And with this simple inequality, we derived an intuition to show why this simple reaction network can have bistable behavior, even though it doesn't look like there is a feedback loop. So the idea is that when the two molecules form two complexes and there is some asymmetry in the system, the two influence arrows we can draw between the two molecules can be adjusted to a feedback-like structure. We then provided evidence supporting this theory. First of all, there are multiple binding sites of miR27 and miR186 at the 3' UTRs of these two genes. And using the in vitro system, we showed that the differentiation of A5 motor neuron is irreversible, and this irreversibility is sensitive to the existence or absence of miR27. And with this theory, we also built a spatial temporal model to explain the data that I explained to you, and we made further predictions, which were then validated experimentally using mouse genetics. And with that, I'd like to thank the students who contributed to this work and our wonderful collaborators, and of course, the financial support from NIH. And I thank you all for your attention.
Thank you very much, Tian. Thank you, thank you, thank you. And for our final lightning talk today, we will be hearing from Kiandokht. I will make sure that you get your slides up. Okay, hello. Okay. They look great.
Perfect. Hello, everyone. Hi, thank you for this opportunity to present at this conference. I'm Kiandokht. I'm from the Mukherjee Lab at Washington University. And today I'm going to talk to you about robustness in organelle size control. So the main question that we want to answer in this project, was how do cells control the size and number of their organelles? And to answer that question, we took a coarse-grained statistical physics approach in which we assume that the cell undergoes these very coarse-grained biochemical reactions for organelle biogenesis. So we assume that we have a cell. It contains a source of organelle building blocks. And from that source, from that pool, organelles can be de novo synthesized at some rate. They can increase in number through fission. They can fuse to each other, and they can undergo decay. And we also assume that at some rate they can grow from that source, or they can shrink. And then to tackle that question, we decided to take advantage of fluctuations in organelle size and number to kind of understand what the mechanism for their control is. And we look at two different sets of fluctuations. First, we look at fluctuations in the average size and number of organelles across a large population of cells. And then to gain some deeper mechanistic insight, we want to look at fluctuations in organelle sizes within individual cells. To do that, what we did is we simulated, came up with a theoretical model and simulated it for those coarse-grained biochemical reactions across a large population of cells. And our focus was really on this this growth process. We solved and simulated for these three different regimes of organelle growth. The first regime is one which is constant growth, in which we have an unlimited source, unlimited pool of organelle building blocks, and organelles can grow at a constant rate. The second regime is one which the cell imposes a negative feedback on what the sizes of the organelles should be. And the third one is one which we assume we have a limited pool of organelle building blocks, and that organelles grow from a limited pool. And what we did was we plotted the joint probability distribution between average organelle sizes and their numbers for these three sets of simulations. And we focus on the correlation between size and number. And we don't really see a correlation in these two first models, but in the limiting pool case, we see this trade-off between size and number. So the more organelles we have, the smaller they get. It's kind of intuitive because, you know, it's like having a limited source of food and then having more mouths to feed. It, you know, everyone gets less the more they are. And then we wanted to compare those results to real life organelle data. So what we did was we fluorescently labeled organelles in yeast cells. We took 3D images of those cells, segmented out individual cells, counted how many organelles they have, measured their sizes, and reconstructed those joint probability distributions for these 4 organelles, for Golgi, lipid droplets, peroxisomes, and mitochondria. And when we look at the joint size and average size and number distribution for these 4 organelles, we again see that trade-off between number and size. So from this, we kind of concluded that these organelles probably grow from a limited pool of building blocks. And this model is actually a very appealing model because, you know, it's kind of appealing to think that, okay, the cell builds stuff until it runs out of stuff to build, until it runs out of resources to build. But there's this very well-known study by the Kandev lab where they showed that when you have Poissonian growth of organelles from a limited pool of building blocks, if you have more than one organelle, if you have multiple organelles, the cell actually fails to control the size of those organelles, and you get huge fluctuations in the organelle sizes. So this led us to measure fluctuations in our organelle data. And to do that, we measured the CV of the organelle sizes within each cell. And we plotted that against the average, we plotted the CVs of Golgi, lipid droplets, peroxisomes, and mitochondria against the average organelle sizes for each. In contrary to what our Poissonian theoretical model predicted, as you see in these insets, for Golgi, lipid droplets, and peroxisomes, we actually see that the CV stays the same, stays constant, no matter what the average organelle size is or how many organelles we have. However, we do see varying fluctuations in mitochondria with varying organelle sizes. So this contradiction between what we expected from our model and the data led us to kind of try and explore an alternative model. So we thought about an alternative model in which we assume that organelles can actually grow from a limited pool of building blocks, but instead in bursts of bursts of randomly distributed sizes, and we assume that these bursts are exponentially distributed, and that these bursts occur at some rate, some frequency of alpha, and at some mean size of beta. And if we assume that in this model that the pool available for organelles to grow from is significantly larger than this burst size, it's been shown that this type of growth can lead to a gamma distribution of sizes. And if we assume that the growth process is regulated by the burst size and occurs at a constant frequency, that we can achieve that flat CV, flat fluctuation profile, because we know that the CV of a gamma distribution is 1 over the square root of alpha. So the next step for us was to test this hypothesis, this new model, against our real data, and we really wanted to do like a parameter free test. So what we did is we, you know, have this like we plot the probability distribution of all of these organelle sizes, and as you can see, it looks like this very like, you know, messy, jumbled distribution. We can measure the alpha and beta for each of these corresponding, for each of these organelles from each of the distributions. So we thought that if we normalize each of these distributions by their corresponding burst size, and then again by their corresponding burst frequency, alpha, that in theory, all of these dots, this size distribution for all the organelles should in theory collapse on this one theoretical curve. So we went ahead and normalized these three distributions, and we actually saw that they do collapse all onto this theoretical curve, completely parameter and fit free. So there's no fitting here. We just calculated alpha and beta from our data and normalized everything, and we saw this collapse. So it's really good news because it could support our model and hypothesis. So just to conclude, what we learned from this project is that these membranous organelles that we studied appear to grow from a limited pool of building blocks, and a burst-like growth model appears to be a good descriptive model for lipid droplets, Golgi, and peroxisomes. And we see that these cells robustly regulate the fluctuations in the size of Golgi, lipid droplets, and peroxisomes. So with that, I want to acknowledge our lab. I worked on this project with my advisor Shankar Mukherjee and ASA, this project was partially funded by the Center of Science and Engineering of Living Systems. And thank you all for listening to my presentation.
Thank you very, very much. Thank you to 3 wonderful talks. That was great.